What if I told you Einstein was wrong? What if the laws of physics were broken by a dead mammoth? Not by magic, not by mutation, but by time itself frozen so perfectly it shattered everything we thought we knew about molecular decay. In the year 2024, deep in the Siberian permafrost, a team uncovered something no one expected. A 52,000-year-old woolly mammoth. Not remarkable for its size or age, but for what was still alive inside it. Chromosomes. Not fragments, not dust. Whole, three-dimensional chromosomes fully intact. This wasn't just a win for archaeology. It was a betrayal of physics. According to Einstein's own 1905 calculations, molecules like DNA constantly vibrate, constantly move. Over thousands of years, those movements destroy the structure. They twist, tear, and reduce chromosomes to fragments. But not here. These chromosomes were perfectly preserved like a sculpture sealed in glass. Scientists call it chromoglass. A state so rare, it doesn't just slow decay, it halts it. The mammoth cells had undergone natural lyophilization, cooling, and dehydration the same way we preserve food. But this wasn't jerky. It was genetic architecture, frozen in time. To test the theory, researchers used real jerky. They blasted it with a fastball, ran it over with a truck, shot it from a gun. On the surface, it shattered. But under the microscope, chromosomes still standing. Their structure, untouched. This meant something bigger. If DNA could survive this way, it wasn't limited to mammoths. It could apply to anything buried under permafrost, from ancient humans to extinct species, maybe even viruses. Structures that should have decayed thousands of years ago might still be whole. And the implications are staggering. Because if DNA can survive in this form, it can be read. It can be studied, maybe even reactivated. But as extraordinary as this preservation was, it wasn't the oldest genetic miracle found in Siberia. That title belonged to something else. Something older than the mammoth. Older than human memory. A fossil, uncovered in the same icy expanse, whose DNA didn't just survive. It remembered a world that no longer exists. And that memory was buried in a tooth. Cold, cracked, silent, until 2021. That year, a team led by evolutionary geneticist Love Dahlen drilled into 3 ancient mammoth molars, pulled from the Siberian permafrost near the Krestovka River. One of them, radiocarbon dated at 1.2 million years old, shattered every expectation. This wasn't just old. It was older than any genome ever sequenced. 500,000 years older than the previous record. A 700,000-year-old Canadian horse. And yet, the DNA was still there. Not perfect. It came in fragments, damaged, degraded, chemically scarred. But those scars? They were the signature of authenticity. Tiny molecular wounds from time and temperature. Proof that this was no contamination. This was real. Ancient. Alive. Researchers painstakingly reconstructed billions of ultra-short DNA strands, aligning them like puzzle pieces, using the genome of today's African savanna elephant as their guide. Slowly, painfully, they built the blueprint of a creature no one had seen, one that walked the earth over 1 million years ago. And when they read the code, they found something strange. This mammoth didn't just carry adaptations for cold fur thickness, subcutaneous fat, circadian rhythm genes. It predated the Ice Age. These cold adaptive traits had already formed long before the frozen world descended, which meant evolution didn't race to meet the cold. It anticipated it, prepared for it. Quietly, slowly, over hundreds of thousands of years. But then came the real twist. The genome didn't match any known mammoth. Not the woolly, not the steppe, not even the Adycha or Chukotka lineages. The Krestovka specimen, so ancient, so silent, belonged to a completely unknown branch of the mammoth family tree, one that diverged more than 2 million years ago. It wasn't just old. It was another. And when scientists traced this lineage forward, they found something even stranger. A hybrid, a child of two species, one of them extinct. The other, legendary. The Columbian mammoth, the giant that once roamed North America, wasn't purebred at all. It was a fusion. But the question was, a fusion of what? In February 2021, researchers at Stockholm Center for Paleogenetics released the results of a decade-long mystery. Their team had sequenced DNA from 3 ancient mammoths discovered in northeastern Siberia. The oldest recovered from near the Krestovka River was over 1.2 million years old and genetically unlike any mammoth ever seen. It diverged from the known mammoth family tree over 2 million years ago, making it not a cousin of the woolly or steppe mammoth but an unknown ancestor. A lineage that left no clear fossil trail, no tusks to display, only broken, weathered DNA whispering its story from the ice. And then came the shock. When they compared its genome to that of the Columbian mammoth, the elephantine beast that thundered across Ice Age North America, something impossible emerged. The Columbian's DNA was split. Not in fragments. In halves. 50% matched the woolly mammoth, a species adapted for cold, known from Eurasia. But the other 50%? It was Krestovka. That meant the Columbian mammoth wasn't a direct descendant of either lineage. It was a first-generation hybrid born from 2 species separated by over 1 million years of evolution. This wasn't subtle genetic mixing over millennia. It was a clean fusion, a genomic partnership, likely forged on the ancient Beringian tundra. One mammoth crossed from Eurasia. One came up from the Arctic. Somewhere, around 420,000 years ago, they met and something new began. No other known Ice Age animal shows this kind of clean genetic hybridization. And yet, the Columbian mammoth thrived. It became one of the most iconic megafauna of the Americas, roaming from Alaska to Mexico. This rewrote the rules of speciation. It challenged the very structure of evolutionary timelines. It proved that hybridization wasn't a rare mistake, it could be a driving force behind success. And just as the idea of ancient animal fusion shook science, another lineage emerged. One not from the Ice Age plains, but from a cave in southern Siberia. A child. A fossil finger. DNA inside. That didn't match any known human. And she would force us to question everything we thought we knew about our own species. That finger, barely the size of a coin, was pulled from the floor of Denisova Cave in the Altai Mountains of southern Siberia in 2008. Just a fragment. No face. No name. No weapons. No tools. And yet, what it held inside changed the map of human ancestry forever. In 2010, scientists at the Max Planck Institute sequenced the mitochondrial DNA from that tiny bone. And what they found was not Neanderthal, not Homo sapiens. It was something else. A genetic branch that split from our lineage almost 1 million years ago, long before modern humans ever set foot in Europe or Asia. They called them Denisovans. There were no skulls to study, no skeletons to reconstruct. Their entire identity was written in molecules. A genome with no archaeological culture, no monuments, no myths, just traces. Yet somehow, Denisovan DNA survives today. In people from Papua New Guinea, in Melanesians, in Aboriginal Australians, and in Southeast Asians. Up to 6% of their genome inherited from a ghost. A people with no face still living in our blood. What's more chilling is what was missing. No stone tools distinct to them. No painted walls. No graves with names. These weren't shadowy figures, they were shadows themselves. Present in genetics. Absent in history. But the mystery didn't end there. Because in 2018, a second child was found in the same cave. But this one wasn't just Denisovan. Her DNA told a deeper, stranger story. One that had never been seen in the fossil record. Not in mammals. Not in humans. A hybrid child born from 2 different human species. One parent, Neanderthal. One parent, Denisovan. And in her blood, An evolutionary collision. This wasn't just a fossil. This was the first known human child born between two extinct species. And what she revealed would unravel the myth of human purity. Forever. Her name was Denny. Not from ancient records from a label. A short, silent tag for a teenage girl who lived over 90,000 years ago in the shadowed chambers of Denisova Cave. Discovered in 2012, her bone fragment looked like the others, weathered, brown, unremarkable. But when her nuclear genome was sequenced in 2018, it sent shockwaves through every evolutionary chart on Earth. Half of her DNA was Neanderthal. The other half, Denisovan. Not distant. Not trace. Her genome was a perfect 50-50 split, proof that her mother and father belonged to two different extinct hominin species. This wasn't long-term admixture. This was direct hybridization. First generation. And it shattered everything science assumed about the barriers between species. For decades, evolution had been drawn like a tree branches diverging cleanly, separating one species from another. But Denny? She was no branch. She was the braid between them. Evidence that Neanderthals and Denisovans, though separated by hundreds of thousands of years, still shared more than territory. They shared blood. Her Neanderthal mother, based on mitochondrial DNA, was from a Western European population genetically close to those from the Vindija cave in Croatia. Her Denisovan father, on the other hand, came from a deeply diverged lineage carrying even older ancestry, possibly from a previously unknown archaic group. In Denny's cells, multiple human timelines overlapped, a convergence of lost worlds. But she was not alone, because in 2019, less than 100 kilometers from where Denny was found, another discovery emerged. Two tiny teeth, belonging to children buried in the high Arctic more than 31,000 years ago. And these children didn't match Denny, or Neanderthals, or Denisovans, or Homo sapiens. Their DNA mapped a completely unknown branch, a people never recorded in any human story. And they were waiting quietly in the Siberian snow. That snow near the banks of the Yana River in northeastern Siberia concealed a grave older than memory. Two molars, left untouched for over 31,000 years, until unearthed in 2001 and sequenced nearly 2 decades later. In 2019, the journal Nature published what those teeth carried. And what they carried was a new name in human ancestry: the ancient North Siberians. Not East Asian, not European, something else. Their DNA split from the West Eurasian branch around 38,000 years ago, before the emergence of recognizable modern populations. They weren't directly related to Denny or even to early East Asians. They were something in between a population caught in genetic no man's land. But they were real. They left signs: carved ivory beads, bone needles, stone tools sharp enough to skin a reindeer, delicate enough to pierce hide. These weren't primitive wanderers. They were Arctic survivors, thriving in temperatures that dropped below -40 degrees Celsius during the last glacial maximum. What shocked scientists most wasn't just their endurance. It was their legacy. Because deep within their genomes was a link to the future. The ancient North Siberians passed on part of their genetic blueprint to the first peoples of the Americas. When humans crossed the Bering Land Bridge between 20,000 and 25,000 years ago, it was the ANS who traveled with them, in blood if not in body. Modern Native American genomes still hold traces up to 40% of their ancestry stems from this lost Siberian population. But here's the tragedy. While their genes crossed oceans, the ANS themselves vanished. No known direct descendants, no living language, no remembered name. They are ghosts with fingerprints. And their discovery rewrote migration history. But it also opened a deeper mystery because another burial site, further south, revealed a child even stranger. His DNA was neither East Asian nor Siberian, but carried signals found in Europe. And that child would connect Ice Age Siberia to the heart of the American story. His name wasn't known, But where he lay told everything. Near the icy shores of Lake Baikal, archaeologists uncovered a grave, shallow, quiet, untouched for 24,000 years. Inside it, the body of a boy no more than 3 or 4 years old, wrapped in reindeer hide, surrounded by ivory swans and a necklace of freshwater beads. Carving so delicate, they whispered care, love, loss. They called him the Malta Boy. But it wasn't his burial that shocked the world. It was his DNA. When sequenced in 2013, the results left scientists reeling. This Ice Age child buried in Siberia carried Western Eurasian ancestry. Not East Asian. Not local. His mitochondrial DNA fell into haplogroup U. His Y chromosome and haplogroup R lineages more common in Paleolithic Europe than in Asia. But the twist was even stranger. He wasn't just European. He was closer to modern Native Americans than to modern Siberians. His genome contained the deep roots of the ancient North Eurasians, a ghost population that no longer exists, but whose legacy lives on across the Pacific. The Malta Boy proved that before the East Asian waves migrated north, Siberia had already been touched by the West, by people whose descendants would someday shape half a continent. Up to 40% of Native American ancestry today traces back to his people. It shattered the myth of linear migration. It redrew the ancestral map. And it revealed a Siberia far more complex than imagined, not just a corridor, but a crossroads. But then came the emotional rupture. This child, this genetic bridge between continents, was forgotten. No oral stories remembered him. No tribe claimed his memory. He was a chapter left unread, buried under snow, until science cracked it open. And if his genes survived the cold, what else did? Because not all things in the ice are dead. Some are only sleeping, and when they wake they don't just change history, they threaten it. The threat came first as a shape, then as a sequence. In 2014, a team led by Jean-Michel Claverie and Chantal Abergel at Aix-Marseille University did what no one had done before. They reawakened a 30,000-year-old virus buried in Siberian permafrost near the Kolyma River. The virus, named Pithovirus sibericum, infected not humans but amoebas. Yet its implications went far beyond its victim. Its genome was massive, over 600,000 DNA base pairs, and nearly 500 genes, with over 90% showing no similarity to any known virus, bacterium, or cellular organism. This wasn't a relative of modern pathogens. It was a biological relic, a giant virus that predated written language, agriculture, and even modern humans. But that was just the beginning. In 2015, they revived Mollivirus sibericum, another 30,000-year-old virus. Then came Pandoravirus yodoma, pulled from thawing wolf remains, dated to over 48,500 years ago, and reactivated in 2022. Each new specimen broke previous limits. Each one infected amoebas, and each one proved that ancient DNA, under the right conditions, could remain viable for tens of thousands of years. By 2023, new classes of viral giants emerged, including Pacman virus and Megavirus mammoth, the latter found near a partially preserved mammoth carcass. And then, in 2025, Finnish researchers announced the revival of Jyväskylä, a genetic anomaly with capsid structure so complex it resembled primitive cells more than traditional viruses. Some of these viruses carry DNA repair genes, immune system modulators, and genomic self-correction loops, features previously thought impossible for non-living agents. These weren't dead codes. These were dynamic systems, and they were waking up. Climate models now show that by 2050, over 20% of Arctic permafrost could melt, potentially exposing millions of dormant microorganisms. Ancient viruses, bacteria, and fungi, preserved in peat, bone, blood, and ice, could re-enter ecosystems without warning. This isn't speculation. It's already happening. In 2016, an anthrax outbreak in Yamal, Siberia, killed a 12-year-old boy and sickened dozens. The source? A thawed reindeer carcass, frozen since 1941, its spores awakened by extreme summer heat. But there's a deeper fear beneath the surface. Because one of the latest recoveries didn't come from an animal, but from a human rib fragment. And what they found in its DNA, no database could explain. But how? How could a human genome buried for tens of thousands of years, frozen, compressed, crushed by ice and time, still whisper its secrets? The answer lies not in the code itself, but in the structure that held it together. In 2024, a groundbreaking experiment changed how we understand DNA survival. At the University of Copenhagen, molecular archaeologists replicated ancient damage using beef jerky dehydrated tissue, blasted with high-speed projectiles and subjected to sub-zero temperatures. The goal was simple: see how chromosomes behave under long-term trauma. What they found was stunning. The jerky samples, despite intense abuse, still held preserved chromatin loops, the architectural scaffolding of DNA. These weren't loose fragments. These were three-dimensional genomic structures, curled and organized, exactly as they would have appeared in a living cell. It wasn't just the sequence that survived. It was the shape. That same year, a permafrost core from northeastern Siberia revealed even more. In mammoth bone marrow frozen for 52,000 years, scientists detected preserved nucleosomes, histone proteins, even trace epigenetic marks. These weren't dead instructions. They were regulated blueprints. For the first time, researchers could see not just what genes were present, but which ones were turned on, which ones might have governed fur thickness, fat metabolism, cold tolerance, which ones were active in death. And if that kind of molecular architecture can survive in frozen mammoths, what about humans? Cairo's preserved mummies, Tibetan monks buried in alpine caves, sealed ossuaries in peat bogs and tar pits. What secrets lie locked in ancient flesh, not as dust, but as code? We are no longer just studying fossils. We're opening living blueprints, genomes that remember, instructions that still know how life was assembled. And there's a reason that terrifies scientists. Because the better preserved the code, the more precise the answer becomes. And there's one question, the deepest of all, that now hangs above every discovery. Where do we truly come from? We are not descendants of empires. We are descendants of whispers. But just as we began to decode the mosaic, one final fragment refused to fit. Because in a preserved specimen recovered in 2023, a sequence was discovered that didn't match Neanderthal or Denisovan or Homo sapiens. And if it's not human, what is it? To answer that, we must first look at what is human and how that definition has already shifted. Since the completion of the Neanderthal genome in 2010 and the Denisovan genome in 2012, geneticists have been redrawing the boundaries of our species. They found that modern humans didn't just coexist with other archaic humans, they interbred with them, repeatedly, across continents and millennia. In 2014, a groundbreaking paper in Nature revealed that the EPAS1 gene, found in Tibetans and critical for surviving low-oxygen environments, wasn't Homo sapiens in origin. It was inherited from Denisovans, a group whose fossils are barely more than a finger and a molar, but whose DNA lives on in millions. Other genes followed. TLR6, TLR1, and TLR10, related to immune function, were traced to Neanderthals, protecting early humans from Eurasian pathogens. The BNC2 gene, linked to skin pigmentation and sun response too, showed archaic introgression. Our variability to adapt wasn't just human innovation. It was inherited resilience. Then came the ancient North Eurasians revealed through the Maltaboy in 2013. His genome showed that Native American ancestry wasn't just East Asian. It was partly Western Eurasian from a population that no longer exists. A vanished people, with no name, speaking through bones in Siberian soil. By 2019, the full genomic analysis of the Yana children confirmed this blend. Their DNA helped explain the origins of Native Americans, tying Siberia not as a bridge, but as a cradle of hybrid identity. And yet, in 2023, a new sequence emerged. Recovered from a human rib found near the Batagay megaslump in Yakutia, this strand didn't align with any known hominin. No Neanderthal, no Denisovan, no sapiens. Its base pairs formed patterns unknown to databases, hinting at a split that predated the known human tree. It contained no modern contamination. It was real, a lineage so ancient it might have branched before Neanderthals were born. And this time, it wasn't just DNA. They found fragments of a skull. And the shape wasn't like ours.